really pleased that Caitlin's giving the second one of these of the two talks this afternoon. And as you heard before in my introduction for the first, these talks are recorded and put on the UK International Coronavirus Network's YouTube channel. Caitlin's going to be talking about the development of a field a test of a LAMP assay for the detection of beta coronavirus 1 in small mammals in Algonquin Wildlife Research Station, Canada, that we've just had introduced to ourselves. Caitlin's doing a PhD at the Liverpool School of Tropical Medicine, and her research focuses on design and development of new diagnostic tests. And the PhD so far is focused on design of low-cost molecular assays to identify key SARS-CoV-2 variants of concern. And she's looking at the use of those assays in Kenya and Burkina Faso. So not going to be talking about that now, but a really nice talk to follow on from Jonathan's. And Caitlin, over to you. Thank you very much.
Amazing. Thank you for the introduction. And yeah, I'm quite glad how nicely this follows on from John's talk. So I'm Caitlin, and I actually recently finished my PhD at LSTM. But I'm going to present some work I did in my lab exchange with Dr. Samira Mubareka's group in Toronto last year, which was in collaboration with John. And this was very kindly funded by the ICN. And this project was to develop a field deployable LAMP assay for the detection of beta coronavirus 1s. So just a bit of background into how this project came about in the first place. As James said, my background is in accessible diagnostics for low resource settings with a focus on viral surveillance. And my PhD, which as I said, I finished recently, was focused on researching alternatives to gold standard diagnostics that could be used for surveillance of SARS-CoV-2 in lower resource settings, primarily in human health. And then I connected with Samira through the Coronavirus Network, and one of the broader projects her group is working on is interested in the investigation of viral zoonosis at the human-animal interface, and specifically investigating the epidemiology and burden of zoonotic disease. And through having some discussions about potential lab exchange project ideas, we started talking about the idea of a lab in a bag. Now, a lab in a bag, we didn't come up with this idea. It's a very popular concept that has been attempted, especially in response to viral outbreaks. We were very interested in applying this to investigating this concept within the context of wildlife surveillance, as there is a need for rapid and accessible diagnostics for wildlife surveillance, and having these tools would allow the development of workflows that allow reliable surveillance to be conducted in almost real time within the field. So ultimately, our project for my lab exchange was to develop and test a LAMP assay for the detection of beta coronavirus 1s viruses in deer mice. And I've just shown a little picture of a deer mice there. And the plan was for me to spend 5 weeks in Toronto. And during this collaboration, we would design and optimize the test, collect the samples from Algonquin, analyze these samples in the field, and then also bring them back to Toronto, where we can extract and further analyze these samples and understand how well the LAMP assay worked. So this is the lovely team I was working with during my exchange. Samira is the PI, and I've shown her in the top left-hand corner. As you've already heard from John, he was the postdoc I was working with during this project. And I also worked with their amazing summer student, Will, who I think is also online now. And then also part of the team was Dr. Jeff Bowman and Lauren Crawshaw, who worked for the Ministry of Natural Resources. So you've already heard a lot about beta coronavirus 1s, but why were we focusing on them? As John said, rodents are a very likely potential reservoir host for these viruses. And beta coronavirus 1 is a sublineage of coronavirus that includes bovine and equine coronaviruses, as well as human coronavirus OC43, as just some of the members. And it's thought that these viruses might have been detected, sorry, let me rephrase that. These viruses have been detected in white-footed mice in Connecticut in the USA. However, not much surveillance until recently has been done in Ontario. And our plan was to develop a LAMP assay that could detect these viruses. So just a bit of background on LAMP for people who aren't familiar. LAMP stands for loop-mediated isothermal amplification, and it's a nucleic acid amplification technique commonly applied to point-of-care testing. It uses 4 to 6 primers to target a region of DNA compared to PCR that typically uses 2 primers. And these massive increase in primers result in rapid amplification of the target region. So on the right-hand side, I have shown a general schematic of the loop structures that form using these 4 to 6 primers. And it is these loop structures that facilitate the rapid amplification of the DNA target. The reaction is isothermal, so it only requires one constant temperature as opposed to having to cycle through multiple temperatures, which improves the accessibility of this assay because you can run it on a heat bath or in a water bath, which makes it more accessible than requiring a complicated thermocycler. It's also incredibly quick, with reactions averaging between 10 to 30 minutes, which obviously is considerably quicker than a PCR assay. Most LAMP assays are also colorimetric, so it can be interpreted with the naked eye, which reduces the cost of the assay as you're not relying on primers and probes. And I've just shown on the right-hand side a picture of a LAMP reaction. So the pink indicates a negative result. And when you get a positive result through amplification, you see a color change into that yellow color. And this color change is due to a pH shift. So as DNA amplification occurs, you get an increase in hydrogen ions, which causes the pH to decrease, and then you see this color change. And another benefit of LAMP is that there is research demonstrating that LAMP can be used on crude samples, meaning that there is the potential for an incredibly simplified workflow that would be appropriate for lower resource setting, where you could just go from swab to LAMP assay to result. So a lot of the LAMP design was very kindly done by John before I arrived, as we wanted to make sure we actually had an assay to be working with for my lab exchange. It was conducted using the N gene of beta coronavirus 1, using representative samples from the different coronaviruses within this species. And then the NEB LAMP primer design tool was used, which is a very easy to use in browser primer design tool. The length and temperature of the reaction incubation also had to be optimized with the aim of minimizing false positives, as these are very common with LAMP reactions. But also we wanted to maximize the speed. So a lot of the optimization work was conducted by Will before I arrived. And his results were that 67 degrees for 25 minutes was the optimum incubation time and temperature for these primers. Another area we were looking at was how to prepare the sample in the field, as we weren't going to be able to perform a normal extraction. This is another area that Will was working on. He was testing a quick extraction buffer, which he was using LCG Biosearch Technologies Quick Extract, which you add to your sample, you incubate for a period of time, and then your sample should be ready to use for PCR, for example. And what Will was doing was optimizing the incubation time as well as the ratio of buffer to sample to get optimum results. And what he found was that a 1-to-1 ratio of sample to buffer gave the optimum results after incubating for 15 minutes at room temperature, and then we would incubate for 5 minutes at 95 degrees to inactivate. So on to the actual sample collection and the research site. The research site we were conducting this research at was the Algonquin Wildlife Research Station, the same one that John mentioned in his talk earlier. And as I understand it, it was started in 1944, and it's just around a 4-hour drive north of Toronto. We're specifically working with the Small Mammal project that was established in 1952, and I think it's one of the longest running small mammal projects in North America. They had approximately 18 trap lines, each with 20 traps along each trap line, and they trap from May to September each year. So while we were there, we were able to sample from 13 different trap lines. We would set the traps at 7 p.m. and then check them 6 a.m. the following morning. We would bait them with soaked sunflower seeds. And then the measurements we took of the mammals were weight, sex, reproductive status, tag number, and then we took 2 oral swabs. We took 2 because we wanted to store them in 2 different types of media, one that could be used for longer term storage and could be used later, and then one that we could use in the field that day. And then once we caught all our measurements, the animals were released. This is our final workflow that we were testing. So as I said, equal volumes of sample to RNA quick extract, followed by 15-minute incubation at room temperature, and then 5-minute inactivation at 95 degrees. And then we could then take the quick extract and add it to the LAMP reaction, which would then be incubated at 67 degrees for 25 minutes. On the right, I've just shown some pictures of our lab setup. So we're very much in a kind of classroom setting. Everything in these pictures we brought with us in the car. So we brought the table, a vortex, and a kind of mini centrifuge, as we were very lucky to have electricity. And then in the bottom picture is a makeshift fridge that I made from a polystyrene box and some freezer packs to keep our reactions cold. And we did it in all weathers. So as you can see on the right-hand side, the lights gave up on us on the last day. So we did this by torchlight. So the results from the sampling, we collected 71 swabs in total. This was over, I think, around 4 days of sampling. 47 oral swabs from individual small mammals. And if we break that down, that's 31 deer mice, 2 red squirrels, 4 jumping mice, and 10 redback voles. And then there were also 24 rectal swabs collected from deer mice, but they were used for later analysis and not for the LAMP. So our results from the field were that we actually got 0 positive results using the LAMP in the field, which is great for the mice, but not so great for our evaluation. All our controls worked, so I'm confident that there wasn't an issue with the assay setup, which is always reassuring. And we kept all the samples and all the quick extract buffer to transport them back to Toronto for extraction and further testing. And here are the results from that further testing. So the top table is showing the results from the quick extract. So this is the solution I mixed the sample with as a sample prep method. As I said, we got 0 positive results from the LAMP. However, when we analysed this mixture with a PanCoV PCR, we detected 2 positive results that were missed by the LAMP assay. This could potentially be a sensitivity issue with the LAMP assay, or as the PCR is a pan-coronavirus assay, it might be detecting coronaviruses outside of beta coronavirus 1. So that needs a little bit more investigation. In the bottom table, this is showing the extracted samples, so on RNA that we extracted from the samples we collected. We then tested these with LAMP and with the PanCoV PCR. The LAMP assay detected 9 positives, but only 4 of these looked to be true positives when compared to the PanCoV PCR. And the PanCoV PCR detected 17 positives that the LAMP did not pick up. But again, it's hard to tell whether this is a sensitivity issue or potentially just detecting coronaviruses outside of the target of the LAMP assay. So the next steps to kind of further understand the results that we got is to sequence the positive samples we got on the PanCoV PCR, as that would allow us to kind of further understand what coronaviruses were present and whether the LAMP assay needs some tweaking to improve the sensitivity, or whether we've just got different coronaviruses circulating within these populations. Doing a limit of detection on the LAMP assay with a designed plasmid control would give us a better indication of the sensitivity of this LAMP assay. And then also investigation into alternative extraction and sample preparation methods would be crucial if we wanted to try this LAMP assay again in the field, as all our controls worked. So I'm pretty confident that the LAMP assay itself was working as it should. We were just struggling to detect any coronaviruses that were there. So potentially we need to do some more optimization of the sample preparation. So what did we learn? The LAMP workflow is feasible in the field. As I said, all the positive controls worked. The workflow was very easy to follow. I think it took maybe 2 to 3 hours max to process and analyze all the samples every day. Like I said, the likely issue is potentially extraction and sample preparation that we would need to look at. But also we were able to further understand the sampling techniques that they use at the Algonquin Research Station. We were able to further understand the diversity of the mammals that they trapped. We set out to just sample deer mice, but on the first day, it was very clear that we were going to be catching a vast array of different mammals. Also the proximity of the trap lines and the ecological differences between them and the number of individuals retrapped within a given week is all kind of data that can be used to further plan more kind of surveillance projects, as this is all information we didn't know going in. And I just wanted to share this little bit of work that's just started and is stemmed from this project. I've now started to look at the application of this same LAMP assay for bovine coronavirus detection, collaborating with a group in the vet school over at Lea Hurst as part of the University of Liverpool. They're currently conducting a study looking at bovine coronavirus transmission within cattle. And I've been able to kind of piggyback onto their study and analyze their samples using the LAMP assay to detect the bovine coronavirus. And the aim of this is to try and use it as a pen-side diagnostic. And the focus at the moment is currently optimizing the use of crude sample. So going straight from sampling the cow to the LAMP assay right next to the pen. That is the aim. And I don't have any results yet. This work is currently ongoing, but hopefully I'll be able to share some more findings very soon. I want to thank you all for listening and inviting me to speak, and obviously thank the ICN for funding my exchange, and to the Wild Epi team, Samira, John, everyone I met in Canada. It was a really amazing experience. Thank you.
Caitlin, thank you very much indeed. That was a really interesting seminar, really great piece of work. So often we focus on the high-tech lab approaches, which mean that that we're missing so many opportunities in low middle income countries, as you know very well. So thanks for that. I wonder whether there are any questions from the floor. So Caitlin, if I could ask you whether you has any sequencing of those products been done at all? Do you have any insight into what may be in the positive samples? Or is it just too early to say?
It's too early to say, I think. The samples, obviously I'm now back in the UK.
Yeah.
So I think the samples are with Jonathan. So I don't know kind of where we got up to with that. But as far as I'm aware, that's like to be determined, which would be very interesting to know.
But I mean, I guess the issue around the potential sensitivity of the LAMP assay, that would be probably no surprise to you, having started that. I mean, you're aiming for detection of more strong positives, aren't you? Rather than ever thinking you're going to get the full sensitivity of optimized molecular detection methods in high-tech labs.
Oh yeah, we would never try and aim to have the same sensitivity as like a PCR, especially with a LAMP assay. I think that would be really tricky. I guess the idea would be kind of use it in a similar way to how you'd use an RDT. It's kind of giving you an indication of what's there, and then you can follow up with more testing. Not so useful with the small mammals, but say with like cows, if you can get an indication of a positive, you can then follow up with more samples, for example. Whereas if you get a negative, you can maybe just leave that animal alone and try another one.
And I didn't pick up, I mean, you mentioned recapture towards the end in terms of kind of further things. Sorry if I missed it. Were you recapturing any of the same individuals over the time course of your study?
Yeah, so we were actually surprised within a week how many recaptures we would get. We wouldn't sample them again, so the team keeps a log of which animals they're getting, obviously. And if we got a number from previous in the week, we wouldn't sample them. But we were shocked at kind of as the week would go on from Monday to Friday, you would see an increase in the number of recaptures. And the team said that this kind of happens every week. You kind of then get a drop off over the weekend and you start again and you get loads of recaptures on the Friday, which is quite interesting because then maybe if we wanted to go back, we might prioritize, say, Monday, Tuesday, Wednesday, because by the Friday, we weren't sampling as much as we were on the Tuesday and Wednesday.
Oh, that's really interesting. That's really interesting. Are there any other questions for either Caitlin or for John? John, you've got your hand up.
Yeah, thanks, Caitlin. Great talk. I was just wondering more conceptually in a way. It seems like the quick extract, the in-field extraction seems to be kind of one of the major bottlenecks. On the one side, you have the technology to support rapid diagnostics in the field, but to get to that point, there's a piece missing. But aside from that, what would you say some of the bigger challenges of in-field diagnostics are?
Mainly, I would say would it come under kind of more logistical? The amount of stuff we had to take, I feel like the dream is to be able to fit everything in a small bag that you could just take with you. But I had a car full of stuff. And initially I think we were like, yeah, we'll do it by the side of the road. It'll be great. But really you can't. You kind of need a room with electricity. So it's kind of the standard problems of like, you need electricity in a cold chain, ideally, to be doing these tests. aside from the fact that, yeah, you're not guaranteed to get enough material in one swab to be able to get the test working on the side of the road. I think I answered your question.
Thank you.
Yeah, thanks, Caitlin. That insight's really valuable. I think sometimes these things are regarded as a panacea for everywhere, and they're probably not, are they? It's I don't think there are any more questions today. Two really great talks. Huge thanks to both Caitlin and to Jonathan for their really interesting talks. That's all for now. There'll be advertised the next in the series coming up in the next few weeks. Thank you both. Can you all thank Jonathan and Caitlin very much for the really two really well prepared, very interesting seminars. And yeah, look forward to the next ones being as good as these ones. Thanks a lot. Thanks to you both, and thank you all for coming. Bye.
Thank you, everyone.
Thank you.
Thanks, everybody. Bye.